All right, let's get straight to it. In this explainer, we are going to crack the code of the Icse biology exam. We're going to do that by revealing the super predictable patterns that are hidden in two of the most important questions you'll face, question 3 and question 4.
I mean, seriously, think about it. What if I told you that for years, the Icse has pretty much been asking questions from the exact same chapters? They're testing the same core ideas, just, wrapping them in slightly different paper each time. By the end of this, you won't just be studying harder. You're going to be studying a whole lot smarter.
So first up, let's decode question 3. Honestly, you should just think of this as the plant world question. Year in and year out, it almost always zooms in on the mechanics of how plants live and breathe, and of course, how their cells divide.
The blueprint for question 3 is surprisingly clear. It always comes back to the same few chapters. We're talking absorption by roots, transpiration, photosynthesis, and the cell cycle, especially mitosis. If you can really master these topics, you've pretty much got this question in the bag.
Now, here is the absolute key. The examiners don't just want you to memorize facts. No way. They want to see if you actually understand how things work. And to do that, they focus on two big things: experiments and processes.
And this is a perfect example. The starch test. It shows up all the time. Let's walk through it. Step 1. You boil the leaf in water. Why? To kill the cells and stop everything in its tracks. Step 2. And this is the one they always ask about. You boil it in alcohol. This is to strip out all that green chlorophyll, because if you don't, you won't see the final color change. And finally, step 3. You add some iodine. If starch is there, bam, it turns a dark blue-black. See? Every single step has a purpose.
Another huge topic is tonicity. You are almost guaranteed to see a diagram of a plant cell in some kind of solution. So in a hypotonic or dilute solution, water rushes into the cell. That process is called endosmosis, and it makes the cells swell up and become turgid. But if you put it in a hypertonic or concentrated solution, water rushes out of the cell. That's exosmosis, and it makes the inside of the cell shrink away from the wall. We call that being plasmolyzed. You have got to know those terms.
Okay, let's switch gears to question 4. If question 3 was all about the plant world, then question 4 is all about us, the human body. This is where all your knowledge of human anatomy and physiology really comes into play.
The big hitters here are always our body's transport and control systems. So we're talking about the circulatory system, the heart, the blood vessels, the whole plumbing network. Then there's the nervous system, the brain, the spinal cord, our reflexes, and finally, the sense organs, with a really, really heavy focus on the eye and the ear.
Eye defects? Oh yeah, that's a guaranteed topic. So you have to know the difference. Let's make it simple. For myopia, or nearsightedness, the image focuses in front of the retina. The fix for that is a concave lens. For hyperopia, or farsightedness, the image focuses behind the retina, and you fix that with a convex lens. So for each one, remember 3 things: the name, where the image forms, and the type of lens that corrects it.
And then we have the reflex arc, which is kind of like the emergency fast lane of your nervous system. It's a simple 5-step pathway you just have to drill into your memory. A stimulus hits a receptor, the signal zips along a sensory neuron to the spinal cord sends a command straight back down a motor neuron, which tells an effector, like a muscle, what to do. That is it. Don't make it more complicated than it is.
All right, time for a little rapid-fire round. Let's just hammer home some of the questions that show up over and over again, along with those perfect ICSE-approved answers. This is pure high-value stuff right here.
Okay, I know we just talked about this, but it is so important it's worth saying again. Why do you have to give that leaf an alcohol bath?
It's all about one key word: chlorophyll. The alcohol removes the green pigment, which makes sure that the final color change from the iodine is super clear and easy to see.
Okay, moving over to the human brain, a total classic, the cerebellum. What's its main gig?
It has 2 main jobs. First, it maintains your body's balance. Second, it coordinates all your muscular activities. If you can write down both of those things, you've locked in the marks for sure.
Next up, the circulatory system. A simple question, but a really fundamental one about our veins.
The answer is all about one crucial job: to prevent the backflow of blood. These little valves make sure blood only flows in one direction, straight to the heart.
And last up for this round, a classic definition question. What is guttation? Well, it's the loss of water from a plant, but not as vapor. It's lost as actual liquid droplets. And the key detail? It happens through these special openings called hydothodes, right on the edges of the leaves.
Okay,
Now for the fun part. Let's look into our crystal ball. We've used an AI to analyze past papers and trends, and we've got some predictions for what's coming up in the 2026 exam. This is your strategic advantage.
For question 3, the AI is pointing to a big focus on experiments and applying your knowledge. So you can probably expect a diagram of a potometer, and they'll ask you about its limitations. You'll also likely need to spot anaphase in a diagram. Just look for those V-shaped chromosomes being pulled to opposite ends of the cell. And keep an eye out for a clever application question, maybe something about why weed killers work better after it rains. And finally, you absolutely have to know the balanced equation for photosynthesis cold. That's 6CO2 plus 12H2O, and don't forget sunlight and chlorophyll over the arrow.
As for question 4, the trend seems to be moving towards little case studies and deeper analysis. So you might get a scenario like, a student can't see the blackboard. You have to diagnose it as myopia and recommend a concave lens. You'll almost certainly see a heart diagram where you'll have to identify something like the semilunar valves and explain why the left ventricle's wall is so much thicker. And lastly, get ready to compare and contrast the sympathetic and parasympathetic nervous systems, like how one speeds up your heart rate and the other slows it down.
So we've broken all of this down, we've decoded the patterns. What does this all mean for you? What's the actual plan? What is your blueprint for success?
Look, the main takeaway is this. The code isn't about memorizing every single word in your textbook. It's about recognizing these patterns that show up all the time. You need to focus your energy on understanding the key experiments, knowing the core processes, and absolutely mastering those labeled diagrams of the heart, the eye, the ear, and the stages of cell division. That is studying smart.
So, there you have it. We've laid out the whole framework for you. The patterns are right there in front of you, and the most common topics are no longer a secret. The real question isn't what to study anymore, it's how are you going to use this blueprint to build your success. Good luck!